So good morning, one and all present here. This winter school session. So today we have Dr. Sakir Ahmed, who is a distinguished professor of rheumatology with around over 16 years experience of clinical and academic experience. He holds MBBS, MD, and DM in rheumatology and clinical immunology and MAMS. Dr. Sakir is internationally recognized for his contribution to rheumatology and immunological research. He also served as a chief editor of immunologic research with a prestigious Springer journal and is also the convener of Digital Health Special Interest Group of Indian Rheumatology Association, as well as co-convener of APLAR, Asia Pacific League of Association for Rheumatology, with more than 185 research publications. 185 research publications is really, you can say, a good number for a doctor who is so busy in our KIMS, Kalinga Institute of Medical Science, and many faculties are giving their interest to working with him. Many faculty of the computer science has also worked with him in many projects. So it is my privilege to get an opportunity to work with Dr. Sakir. He is a highly sought after speaker and delivered invited lectures at global platform, including Washington DC, Singapore, Japan. So really, it's an honor to us, Dr. Sakir, you are with us. And definitely after the winter school also, as I have promised, we'll do some good work, which will be and definitely helpful for our society and our research communities. So thank you, sir. Now, sir, session is yours. Thank you very much.
Thank you, sir, for the kind words. It is indeed an honor to be invited to talk on this. And uh, I'd like to share my slides. And if anyone has any doubts, please interrupt me at any time. I'm happy to reply to any questions. So I'll be talking on systems biology and precision medicine in rheumatology. As sir has told, I'm the chair for both Asia Pacific as well as Indian Rheumatology Association Special Interest Group on Digital Health. So something regard to AI, and this is something close to my heart, and we have done a couple of projects on AI. But before going to that, I'll just tell you what is rheumatology. Like people have limited understanding something rheumatology is like rheumatoid arthritis. So it is a subspecialty of internal medicine. Once you have completed your MBBS, then your DM, or then your MD or DNB in medicine or pediatrics. After that, you can select subspecialization as a doctorate in rheumatology. So what does rheumatology deal with? It deals with all kinds of arthritis and musculoskeletal problems, plus multisystemic autoinflammatory or autoimmune diseases. You know, where the immune system is not working properly, they can cause problems in the kidney, in the heart, in the lung, and they can also cause arthritis. But if it is more than one organ involved, then you need to know who is a rheumatologist. Then what exactly is systems biology? So there is a definition, holistic qualitative approach to study complex interactions, etc., etc. But very simply put, the study of the DNA is genetics. And the DNA is converted into RNA, and that RNA is converted into proteins, which do all the works which make our body. They are the structural and functional components of human life. So the DNA is study of DNA is genomics. When it is transcribed to RNA, it is called transcriptomics. When it is translated into proteins, the study of the proteins is called proteomics. And the proteins will have so many chemical reactions in the body, so many small molecules are involved, like fatty acids or different types of sugars, which keep the cell running from day to day, from second to second. If we study those biochemical molecules, this is called metabolics. And ultimately, the manifestation of all the protein, the metabolomics, they make an individual. And the functioning of that individual is the phenotype, which may be in health or in disease. So these are the various levels of which we consider when studying a biological system. And when you integrate all of this, it is called systems biology. To make things simple, there is an Indian dish which is called khichdi. You mix everything up. It is basically systems biology. So just to understand, genomics will have multidimensional data. For each patient, you will get 30,000 genes. Transcriptomics, you'll get 50,000 RNA transcripts for one individual. For proteins, again, the number is proportionately higher. And when you're talking of systems biology, you have interactions between 30,000 into 40,000. So when we are talking of systems biology, the big data is raised to the power n. If you are taking three, genomics, transcriptomics, proteomics, your amount of big data is exponentially raised to the power 3. So when we want to do something like the systems biology, conventional statistics may not be very helpful, and then we have to use something like AI. I'll give an example of our work done. I am working on one disease called reactive arthritis. What is reactive arthritis? Suppose you have diarrhea or you have an infection in the urinary tract, like the urine burns, there is high grade fever. The infection, you take some antibiotic or whatever, it resolves. After 2 to 4 weeks, there is a development of arthritis. So the antibodies or the immune reaction to this infection leads to an arthritis which may affect your knees, your peripheral joints, or it may affect your backbone also. So this is called reactive arthritis. The bacteria is not going into the joint, but the immune reaction is causing the inflammation in the joint. So we have done genomic work. There is a HLA B27 gene which is associated with disease. So we have done subtyping, what polymorphisms are there. Then we have looked at the cytokine profiles. So what I have told is like genomics, transcriptomics, then there are others also. It is not just five of them. If you look at the cytokine profile, there are hundreds of cytokines in the body. So if you look at the cytokine profile, you call it as cytokinomics. You can look at the methylation pattern of the DNA, that is called methylomics. So there are so many more omics in um, systems biology. So we looked at the cytokine, what cytokines are, the chemical messengers which lead to inflammation or resolution of inflammation. Then this was in the serum or the blood. Then we took out the fluid from the knees, which is called synovial fluid. From the swollen joints, we have taken out the fluid. Again, we have done the study of the cytokines. And if you see conventional statistics, when we use, we do not find much difference. But when this is multidimensional data and we really want to process it, we need some kind of AI or at least machine learning. Then we have worked on metabolomics. We have taken the blood and synovial fluid from the joints of patients with inflammation. And we have used nuclear magnetic resonance to find out what molecules are there. And you'll get something, you can get what is a spectrogram. One is for the healthy controls, one is for patients with reactive arthritis, and one is for patients with rheumatoid arthritis. Rheumatoid arthritis we have taken as positive control. NC means normal control, RA is rheumatoid arthritis. This is the positive disease control and reactive arthritis. And you can see there are thousands of molecules. We can expand this and look at each molecule, and this will give us the concentration of each of the molecules. And again, the basic thing what we can do is a PCA, which is again, you'll know the basic unsupervised learning, and we can find out the variables which are important for projection. There are different kinds of uh, two-dimensional analysis. This is the orthogonal LDA. Again, we can use all these statistical tools. We can use the K-means clustering and so on to find the different metabolites. Then instead of looking at individual molecules, we can look at a set of molecules. And when we look at a set of molecules, we can say which pathways are upregulated. So this is the same metabolomics data we have. We have seen that in patients, cysteine metabolism, citric acid cycle, fructose. So these are the specific pathways which are upregulated or downregulated in patients having this disease. So these again will require some kind of advanced statistics. The next thing we have done is the metagenomics. Genomics is the DNA that we have. But do you know in our gut, there are 10 times more bacteria than cells. If total in our body there are 10 to the power 8 number of cells, there are 10 to the power 9 number of bacteria in our gut, on our skin, everywhere, interacting with us day in and day out. And if we take a sample from the gut and we do what is called next generation sequencing, we are able to identify all the bacteria in the gut. This is called metagenomics. Meta means beyond. So it is the genomics of all the common cells, bacteria which are living along with us on in our gut or our skin. So there are again millions of different species. Most of them are non-culturable, so we have to do the sequencing and identify them. And again, here also we cannot use conventional statistics when you have so many thousands of molecules. You need to do dimension reduction. We have done that. The standard procedures are looking for alpha diversity. Again, you can see there are 5, 6 different methods. Then beta diversity, we can look at using NMDS. Again, these are statistical methods. Then we can look at individual phylum. If you know the division of classification of life, highest is the kingdoms, then phylum, then you have family, then you have genus. I'm not going into details what are our findings, but I'm just showing how these things are being done. And ultimately at species, we have identified like these are the 8 bacteria which are associated with reactive arthritis. Then we confirm by another statistical method. Since this is big data, doing by one method may not be sufficient. So we have to do another method and confirm that we are getting the same results by the other method also. Then again, finally, we look at them individually and also see if they are statistically different with false discovery rates. Correction for p-values. Then this data we have gone ahead and applied machine learning, basically support vector machine. So we have taken the gut microbiota at the baseline and we have followed the patients for 3 years. And we have seen that how many of them become chronic and how many become disease-free. We had approximately 50% who had become chronic and 50% who were disease-free without any drugs. And we have divided these patients into a training set and validation set using support vector machine. And we have used some clinical parameters plus the gut microbiota. And accuracy was not that high, but we got an accuracy of 0.8. And we found out which are the species or genera which is important in the prediction model. So possibly if these blue ones are present, then the person is more likely to go into disease-free state. And if the red ones are present, these patients are more likely to go into a chronic arthritis. This was presented at American College of Rheumatology by our student, and it was projected. They had a spotlight on their website for almost 1 year. That this kind of work is also being done. Then I presented it Doppler Asia Pacific, and this was awarded the best abstract award there. Next, what we have done this year is transcriptomics, that is the RNA. Since for each gene, you can have 10 splices of RNA. So the number of data increases again 10 to 100 times. And there's technically different things. So we have used something like KIMS clustering. And we have shown again what is the gene expression of reactive arthritis who have become chronic versus those who are in remission. And we have also identified single genes, as well as I have shown, we have done the pathway analysis. So if Shigella, there is a reaction to Shigella infection, reaction to Salmonella infection, they are more likely to go into remission. This is one is from peripheral blood, and the other is from the fluid from the inflamed joints. Then we can go one step further and combine all the pathways and do a network analysis. This has been presented both at UPLAR and ACR by our students this year. So what we have done in reactive arthritis, we have done the clinical profiling, the genomic profiling, the cytokine profiling, metabolic profiling, gut microbiota, and transcriptomics. So at 5 different levels, we have explored the disease. And now when we want to do it across all the 5, then definitely it's the systems biology we have to use. higher statistical models, basically what we are calling AI. So what is systems biology? It is a hypothesis-free approach. You do not have a, you just get the big data, you correlate and you find out that what is predicting the outcomes in the patient. Best is if you can show similar things at the gene level, at the RNA level, at the protein level, and the metabolic levels. But again, the amount of data is very, very high. And again, we have to validate also. And this is the basis for precision medicine. What is precision medicine? Can you take a sample of blood and tell what is going to happen to this patient after 3 months? Patient will be perfectly fine or patient will develop some complication. From the blood, can we predict this person will be benefited more by drug A or more by drug B? Or side effects? Can we predict that side effect is more likely with drug B than with drug A from this baseline multiomics data? So that is the holy grail of personalized medicine. So you just take the sample, you look at the DNA, you look at the RNA, whatever, and you predict what drug is going to be best for that person. And also you can tell him that this is likely that if there is 80% probability you'll be disease free by 2 years. So that is what we are aiming at. And since I have time, I'll just show a little bit of other work we have done. Since this winter school is focused more on imaging, we had done some imaging work in CTD-ILD. CTD-ILD is like basically autoimmune ILD, where lung disease occurs because your immune system is hyperactive. So that is called CTD-ILD. In CTD-ILD, we took the CT scans and we tried to predict that whether what is the risk of infection in this patient. Can the CT help us predict it? So these are the inclusion exclusion criteria. I'm not going to details. So basically we had clinical features. We had around like 17 clinical features, which we used for a neural network. And for the imaging data, obviously you would be knowing we use a convoluted neural network because the spatial difference between the data is also important. It is not just the data, it is how the pixels are placed relative to each other. And we tried to build a model. We had 54 patients of CTD-ILD. Unfortunately, our patients of rheumatology are very limited. You will not get 100 patients, but 54 is a very good number of patients. But for conventional statistics, this 54 will not give very good answers. So definitely, when we want to uh, do this type of analysis, we are completely dependent on AI. So different patients we followed up approx just less than 1 year, and there were 38 instances of infection in these people, and these were the drugs used. So what again we divided 10%, 90% for training and 5% for validation, and the itinerations were repeated 10 times over and over again, and this was the final. We got a median validity of 98%. This is obviously internal. We have to validate it externally. And this is the example. If patient one is given one drug called azathioprine, the risk of infection is around 30% in the first 1 month, then it gradually comes down. This is also same for most of the other drugs. This is again patient number 5. Again, his profile is different. His risk of infection will increase after the 6 month. Initial 3 months, the risk of infection is low, but his risk of infection is likely to increase after 5 to 6 months. So again, this patient number 10, his risk of infection is almost zero. So this information is clinically very meaningful to us. And if we look at different drugs, they are following almost a similar pattern. So maybe the initial 1 year, the difference, the risk factor for infection is not the drugs, but the basic how much the lung is already damaged. But if we look at different drugs, of same drugs in different patients, we can see how one individual differs from other. So the risk is you cannot have a same risk for all patients. These three, 1, 2, 3 patients, they had high risk at beginning, then it plateaued. Then this few patients had high at the beginning. One patient probably had high after 6 months. So every patient behaves differently, and this is what we are trying to predict. So this is the only work we have done using image-based. We have already used the LLMs for a clinical decision support system like diagnosing rheumatoid arthritis. Initially, we had 160 patients, but now we have around 320 patients, and we call this SARA, screening agents for rheumatoid arthritis. We use agentic frameworks, solo agent, dual agent, as well as higher agents. The higher agents did not work so well, and the trio did not work. So most of our data we have shown on with solo and um, trio agents. So the diagnostic accuracy in using Gemini, using DeepSeek, using Llama, you can see most of the models are performing very good. And now we have actually updated, we have 320 patients. The accuracy is around 90% for most of the models, but there are certain issues, what we call the mm, reasoning. The LLMs give you the correct diagnosis, but it cannot tell you why this diagnosis is given. When we ask for the reasons to why this diagnosis have been given, so the LLMs are not able to explain, or they are giving all the wrong explanations. So explainable AI is something else we are looking at, because until the clinician knows that the LLM is reasoning properly, no one is going to trust these results. This is a confusion matrix. So this LLM work and um, CTDILD work that was done with the help of Dr. Murari from computer engineering, and however the basic work using support vector that was done entirely by myself. These are my students and one of Dr. Murari's students. So thank you. I think I finished before time, so we can allow any questions if possible.
Thank you very much, sir. Many of our CSE students and not even we also know that you have already implemented so many things, Agentic, LLM, many things you have already used. So really, it's great, sir. And from the presentation, I come to know that we can have many problem statements in which students can also work. As I promised, I will ask the student to join and do some prototype, then only we can have some project proposal where
Yes,
student
sir.
also can be worked. So thank you very much. And Sakir, Dr. Sakir has already seen 15 patients in OPD. So from their busy schedule, he had given us some time. So it's really great. Now it's open for all the students. They can ask the questions. You can unmute yourself and you can ask. Yatharth, yes.
Am I audible?
Yeah.
Yes, yes.
So
Yatharth.
hello.
Yeah,
Yes, sir.
Yatharth. We can hear you.
I think the way you have used like the gut microbial systems and the different type of biomarkers, it is actually very creative. But one, I would say fallacy with that thing, which I think may arise, is that it a model which is trained on like an Indian population data set on these biomarkers may not work for someone in Africa or may not work with someone in America, because they may end up having different immune responses and they may end up having different gut systems due to like their different diets. So how actually exaggerated is this issue, or is it not exaggerated, and how do we overcome it apart from the simple key get a larger data set, because that is a very expensive answer to that question.
I think you have answered your question. This is something we all know. GIGO. The best models cannot be better than the data you have. And if I'm modeling here on some Indian microbiota, I cannot obviously extrapolate that to something which is in Africa. But fortunately or unfortunately, reactive arthritis is a local problem. If you go to Europe, you will not get reactive arthritis except maybe a little bit in Finland. So I have taken up reactive arthritis as somewhere to work because it is so much predominant in the eastern coast of India. So I think we have to keep the global perspective, but we should also focus on local problems. Yes, once we have a model, we are able to validate it or we can improve it from other data sets. It is great, but except Mexico, maybe Finland, most other countries do not see the reactive arthritis that what we are seeing.
So for most of the diseases and issues in general, which biomarkers are the most constant that we can focus on if we are creating a global product?
See, most of the big data you have seen in the last 10, 15 years, so much of big data has come in. But the problem is that everyone is sequencing differently. The controls are different. The analysis may be different. There are so many different softwares also. And even when you are using AI, there are so many different types so you can use. So there is a lack of consistency among studies. Now, as the amount of data is growing more and more, we are having international collaborations. Now we are seeing certain trends, but still that precision medicine is not here. We have to work maybe another 5 years, 10 years, and our main work is not the models. We are making very good models, but our main bottleneck is the quality of data we have. Once we have pure their data over time, then will whoever has the purest data is going to win this race.
Understood. Thank you.
Okay, any other questions? Abhishek, yeah.
Since you have the experience, I would like to know if you have some tips or something that you feel that can be avoided or we should not do for making the prototype. Like if you have failed something that you should not have done in the early stages of making these models.
So what I'm seeing is most of the people they are using cross-sectional data. Means today patient has come, patient has joint pain is this joint, and you have this data. So cross-sectional data is not very good for modeling. If you want modeling, you need to have longitudinal data. Means patient has come today, these are the parameters, and after 6 months, after 1 year, what has happened? Then there will be predicting power. We need models who can predict which are, since you are talking of marketable products, we need to have longitudinal data used for the predictions. I hope that answers your question. Most
Yes,
of
sir.
the computer science people, 99% whom I have met, they are trying to work with cross-sectional data. Shift over from cross-sectional, go to longitudinal data.
Okay, okay. Thank you, sir.
So one point from my side. So many of our students, computer science students who don't know biology, they're afraid to work on the system biology or biomedical. So I can say them that one book is their mathematical biology. So where biology is not, that deep knowledge is not required. Everything they are related with the mathematics. JD Murray, some book is there that two versions are there. Two volumes are there, one and two. Just go through that one, then definitely will contact with sir. And I thought that so many problems you can derive from here. Yes, anyone? Somya, yeah. Continue.
Good morning,
Somya,
sir.
yes.
Good morning, sir.
Hello.
Good morning, good morning.
Sir, I want to ask, like, if you are prescribing like a patient on the basis of an AI model, like we are using a LNM and genome sequencing. So, like, if the problem is according to another disease, like you are saying the arthritis, so the trial and error method is must be using only on that. Like we are increasing the doses by one to see the result. So don't you think it will not work on everybody, like the trial and error method of giving the medicine?
That is the best model we have that is working till now. And if we want to refine it further, then we need to go for that personalized medicine or what we call precision medicine. And for that, we need data. You give 100 people the drug, you follow them up, you see their genetics, their RNA, their DNA, whatever, and you build good models. Then you can go back and use those models to use the precision medicine next time. So until you collect the data, the models are very useless. That is what
Okay,
I'm saying. We need to have good data, follow the patients up at least 1 year, 2 year. Then from the data, when you model, you'll get correct answers.
okay, sir.
Yes, Dr. Ponapa, any question from your side? Sir, Dr. Ponapa is working as an assistant surgeon. But after that, he's doing his MCA in AI and data science. So yes, Punapa, Dr. Punapa.
Good morning, sir. I think
Hello.
today's discussion and presentation, I think it has given me a lot of insight into actually how we can integrate medicine and AI. All these days, I was just sort of through a tunnel vision. Okay, medicine and computers, okay, people are talking. But now today's presentation, I can relate very much about how to use our data. I am doing some works on diabetic foot and all those things. That is where I got interested into this AI. Thank you very much, Dr. Sakir, for I think igniting more passion to involve myself with the AI. Thank you very much.
Thank you for the kind words. I think the dialogue is needed. We are scared of the computer science jargon. You may be put off by the medical terminology. But once we start working together at an individual basis, then we'll understand better and we'll get more ideas.
I would agree on that. That's why I think now this is the beginning. I met Dr. Satyaranjan Das, and now this is the beginning. I hope so. And you also. Thank you.
So last question, Sudhanshu sir. Do you want to ask any question, Sudhanshu Patra? Any
No,
question
from my side, there is no question, and I should appreciate Dr. Samir because the way
Sakir.
he has presented the topic with computer science. Because I have asked, you said I am also here in biology. So understanding all the things is little bit difficult for me also. But yes, I have to work on this area on biology and this medicine so that we can do some work in medicine with computer science. Thank you.
Sudhanshu sir, that book you just follow, Mathematical Biology by J.D. Murray. Two issues are there, volume 1 and volume 2. Go through that one. Definitely it will be helpful and so many innovative work you can do. You just go through that book first, complete that 2 books first. I'll provide you that soft copy is there with me. So definitely.
Okay,
Thank you, Dr. Sakir. Very
Thank you,
informative
everyone.
and nice presentation. Definitely, I'll be in contact with you. Thank you very much. So I
Thank
think
you.
next section will continue with N.Sagarika of Gynec department. Thank you, sir. Namaste, sir.
Thank you, sir.
So we have the next session of Dr. N.Sagarika. Ma'am, are you here?
Yeah.
Hello.
Yes, good morning, sir. Yes, I'm there, sir.
Yes. So now the next presentation is from Dr. N.Sagarika, who is working. I think he's the MD. She's doing her MD in Gynec department of O&G. So she's going to give the presentation. Yes, ma'am.
Sir, I'm working as assistant professor in the department of
Sorry,
Gynaecology.
very sorry. So she is working as an assistant professor in our Kalinga Institute of Medical Sciences. So when I had visited to Dr. Pamila, she only proposed the name of Dr. Sagarika. She is working in various projects. I think 2-3 projects she has already engaged herself. So very sorry, Miss, I have not mentioned.
Okay, sir, fine, sir, fine.
So now she is going to present on ovarian morphology and ovarian artery Doppler indices in predicting fertility outcome and metabolic consequences in PCOS. So thank you, ma'am. Session is yours. Once again, I'm very sorry I have not mentioned.
Okay, sir, fine. Thank you so much, sir.
Okay. Thank you, ma'am. The session is yours. You can continue.
Yeah.
I've already made you the co-host. You can share the screen, ma'am.
Yes, sir.
Yes, ma'am. Your slide is visible.
Yes.
You can make it full
Okay.
screen.
Okay. Good morning, everyone. Myself, Dr.N.Sagarika. I'm working as assistant professor in Department of Gynae, KIMS, Bhubaneswar. Thank you, sir, for giving me such a nice opportunity for this winter session. Today, I'm going to discuss my topic, ovarian morphology and ovarian artery Doppler indices, predicting fertility and metabolic outcomes in PCOS. My agenda of discussion will be something about PCOS, what is PCOS, the basic knowledge, then what are the diagnostic criteria, then what are the Doppler ultrasound findings in PCOS compared to the normal ones, what are the fertility outcomes and the metabolic complications in PCOS, and in the last I'll discuss few points about how AI can help us in the diagnosis and management of PCOS. PCOS is polycystic ovarian syndrome. It's a multifaceted endocrine metabolic disorder, basically, and the trend is rising very much currently among the reproductive age group. It can occur in the adolescent group, like 15 to 25, also in the reproductive age group, and it can also occur in postmenopausal. But the main age group is the reproductive age group, that is between like 15 to 45 years. And there are many why I have chosen this topic is because because of the sedentary lifestyles, the dietary intake of today's generation, and the obesity. All these factors lead to PCOS. So basically, here it occurs some endocrine dysfunctions like hyperandrogenism. That means there is increased level of testosterone, that is the male hormones. There is imbalance between the female and male hormones that lead to all these manifestations of this syndrome. And another characteristic is the menstrual irregularity because of this hormonal imbalance. So ultrasound, that is the imaging, plays an important role, a vital role in the diagnosis and management of this disease. So basically what happens, there is androgen, that is the male hormone is there, testosterone, and female hormone estrogen is there. Because of excess of estrogen, that lead to excess of androgen production and insulin resistance and all the complications of PCOS. So these are few symptoms. The main symptoms will be the menstrual irregularities. Second features of hyperandrogenism, that it will cause acne, hirsutism, all that. Another fertility complication, because there will be no ovulation in this case, so leading to infertility issues. Another complication is weight gain. So obesity is an independent factor. It can lead to PCOS. Also, PCOS can cause obesity. So there are so many diagnostic criteria in PCOS, but the main diagnostic we follow is Rotterdam criteria, in which it has taken 3 criteria into account. One is the anovulation, that means absence of ovulation, for which the patient will be having menstrual irregularities. Second feature is hyperandrogenism, that because of the increased male hormones, there will be features like acne, hirsutism, that is excess hair growth in the body. Third feature is the polycystic ovary morphology. It's an ultrasound finding, that is there will be polycystic ovaries will be seen in ultrasound. So out of these 3 criteria, at least 2 criteria should be present as per the Rotterdam criteria. There are 2 types of PCOS morphologies. One is non-PCOM type, one is PCOM type. In PCOM type, there will be polycystic ovaries will be there. Because of that, menstrual irregularities will be the predominant feature. But in case of non-PCM, like some patients will, in ultrasound, the report will be normal, but they will be having other features like excess testosterone levels, that is hyperandrogenism. And in these cases, metabolic risk will be more. This is the main 2 differentiation. So in ultrasound, the recent guideline has come in 2023, ESHRE. In the ultrasound, we have seen the number of follicles. See, in the ovary, every month, in the menstruating lady, 1 follicle ruptures and ovulation occurs. But in case of PCOS, this number of follicles will be more. It won't get matured, it won't get ruptured, so that ovulation cannot occur. So these immature follicle number increases and seen in the ultrasound. So as per the recent guideline, in transvaginal ultrasound, we can see about more than 20 follicles per ovary, or the ovarian volume we calculate from the length, breadth, and the width of the ovary. Basically, the ovarian volume will be more than 10 ml. Similarly, in the trans-abdominal ultrasound, we will see the number of follicles per section of the ovary, which should be more than 10. And also the volume will be more than 10 ml. So I show a picture that there will be peripheral arrangement of these all immature follicles, giving it a string of pulse pattern. That is the characteristic features of PCOS. And also we will see the stromal echogenicity and the volume. That means the normal structure, the glandular structure of the ovary will see, it will appear more echogenic. It's a basically ultrasound finding. So we can see in this picture, this is the peripherally, there are so many more than 10 follicles are there. We can see it's called the pulse string appearance of the follicles, and also the stromal area. When we take the ratio of the stroma and the cortex is the part of the ovary. When this ratio is more than 0.32, we tell it is mostly a case of PCOS. So we have to see how can we correlate the ovarian morphology and fertility. In case of PCOS, there will be increased number of follicles that reflects that there is an ovulation. That means there is no ovulation. And the ovarian volume, when it is more, when the stromal echogenicity is more, it means there is a hormone level, like anti-Mullerian hormone and luteinizing hormone. These hormones levels will be high. So it will predict that the patient may have infertility issues. And another thing is there is inverse correlation between ovarian volume and induction response. That means when the ovarian volume is high, when you treat the patient for infertility, the response will be less. So it's an inverse correlation. And after ART, that is artificial reproductive treatment, the response, there is a case like hyperstimulation. The ovaries will be hyperstimulating, but the outcome will be the outcome of clinical pregnancy will be less. So we can see in this picture, this is a basic picture of showing uterus and ovary. Basically, ovary, it is supplied by the ovarian artery, which is a branch of abdominal aorta, and it also gets the blood supply from the uterus. So I have shown you the ultrasound picture of ovarian artery, which is the main point of our study. In the ovarian artery, by doing the Doppler, basically we see what are the changes in the ovarian artery during the systolic and diastolic phase of the cardiac cycle. So we will get some waveforms like systolic waveform, diastolic waveform, we can see in the picture. And from that picture, we will calculate some factors like basically two factors we see, PI, that is pulsality index, and resistive index. So there is a calculating formula is there. PI is calculated by systolic minus diastolic divided by mean, and RI is calculated by systolic minus diastolic divided by systolic. So basically it reflects the blood flow. What is the blood flow in the ovarian artery? What is the blood flow and the blood flow velocity to the ovaries? So with that we get this waveforms. Now what changes occur in PCOS? What happens? The normal range of this PI and RI are like PI is between 2.2 to 3 and RI is between 0.6 to 0.8 in normal ovulating patients. But in PCOS, what happens? Because of the high echogenicity, there is high blood flow. It reflects that there is high blood flow, means there is less resistance in the vessel. So these resistive indices values will be comparatively less. So RI and PI values will be less only in case of PCOS. And PSV is another factor, that is peak systolic volume. It will be increased. That means there is high blood flow. It indicates that there is hypervascular stroma in the ovary in case of PCOS. And also this is a 2D ultrasound. In 3D ultrasound, we can see other factors like VI, vascularization index, FI, flow index, and VFI, that is vascularization flow index. These factors will be increased in case of PCOS. So now we basically, what are the complications in PCOS with fertility? Normally, because of an ovulation, that is, it will lead to mostly infertility issues. If at all the lady gets pregnant, then there is high risk of miscarriage rate, and the number of live births, that is completion of the total pregnancy number, is lesser compared to the general population. So how Doppler helps in predicting these infertility issues? So we will study and PCOS can be ovulatory also or in anovulatory. Like in non-PCOM cases, ovulatory PCOS will be there, and those who are anovulating, in these patients, the PI and RI will be elevated throughout the cycle, and the resistance ovarian artery does not change, and high vascular resistance contributes to the follicular arrest, because so many follicles are developing at one time, and the vascularity is very much high. So the single follicle that is required for ovulation is not available. That leads to infertility. But in other case, like in non-PCM or ovulatory PCOS, there will be normal ovulation, and just before ovulation, PI and RI will decline. And these Doppler indices will also help us or guide us in during the treatment or management of the PCOS, because with treatment, these values will change and get normalized. So help us to know the success of our treatment. Then comes to our second point of interest, that is, what are the metabolic consequences in PCOS. So basically here, because of imbalance of the all hormones of the body, it predisposes to some metabolic diseases like diabetes, hypertension, or any cardiovascular high risk diseases will be there. And from some blood tests, so we can know what are the, we can predict, like in case of diabetes, there will be, we'll see along with fasting and PP, we'll see fasting insulin level will be increased. Similarly, in cholesterol levels, LDL levels, this is for lipid profile, will be de-risked, and the good cholesterol, that is HDL level, will be low. So all these complications may arise in PCOS. So how, again, Doppler indices, doing Doppler, it can predict metabolic correlations. Few studies have been done that it has been shown that low RI and PI will correlate with high fasting insulin and home IR, as a test for diabetes, and it is linked to androgen excess and insulin resistance. Insulin resistance means there will be secretion of insulin from the body, from the pancreas, but it won't act at the level of the cellular level, and ultimately leading to the hyperglycemia, that is the diabetic status. But with treatment like metformin, so treatment for diabetes, again it will improve. So by doing the Doppler indices, we can predict the metabolic risk factors for that patient. Also with treatment, whether she's improving or not, we can also predict that. So my area of interest will be like how AI can help in diagnosis and management of PCOS, how it can help in predict the all these complications, especially the fertility outcomes and the metabolic consequences in PCOS. We can study for about, like, we can study in the reproductive age group between like 20 to 35. For 5 to 10 years, we can study the patients of PCOS. Then we can just observe, and we can see what are the complications, like metabolic complications, or whether she is having some infertility issues. We can study. So AI in many ways can help us in this diagnosis and management, like the better development of the ultrasound machines like 2D, 3D, and 4D. Second thing, AI can precisely help in counting the number of during ultrasound, number of follicle count, better delineation of the stromal echogenicity, the proper texture quantification can be better done by developing some AI models. Third, we can see it can help also aid in the diagnosis of this PCOS. Like what we are going to see the number of follicles per ovary, per section of the ovary, what will be the ovarian volume, and Doppler indices, and the proper vascular mapping can be well precisely done by some AI models. So it's basically machine learning and deep learning will be there. And we will correlate the fertility outcomes and the metabolic risk factors, including all these ultrasound findings, Doppler findings, the clinical feature of the patient, and the blood hormones level. All we can correlate to get final conclusion in the diagnosis and the management. So, at automated imaging and AI can reduce operator dependence, because when individually we are doing that, there may be differences in findings. But when we can use the AI model, the operator dependence will not be there. So very precise and accuracy of the testing will be more. And in that, we can take all the factors like morphological factors, the hemodynamic or the Doppler findings, and what are the metabolic data from the blood investigations. So that will help in precision in the diagnosis and also management. So my future project proposal for AI in PCOS imaging will be that it can also help in a data collection from various institutes or clinics, or from the ultrasound and Doppler data sets we can collect, which will be a homogeneous data will be provided. Second thing, it can help in algorithm development, that will help in the classification of various models for ovarian morphology. And the pattern of the Doppler indices, Doppler signal pattern is very important. We can, like in uterine artery Doppler, we get a specific diastolic notch. But in ovarian artery, we have not seen like that since far till now. But by studying in a long term, even we can, it can help us to recognize any specific Doppler pattern in ovarian artery. Third thing is it can also help in developing a model that can predict the fertility outcomes and the metabolic outcomes. So combining imaging with hormonal and metabolic data, it definitely will help us. And it should be a user-friendly tool for all the clinicians and researchers, so that in every setup it can be easily accessible and useful. So interdisciplinary collaboration between biomedical scientists and engineers is essential to realize AI-enabled precision diagnostics and personalized therapeutics. So my conclusion is that ovarian morphology and these Doppler indices provide a complementary insights to PCOS pathophysiology and definitely fertility and metabolic risk factors. We have to see all the metabolic parameters like FNPO, ovarian volume, and vascular indices like PI, RI, and PI, FI, all that. That will help us in phenotyping the type of PCOS also. So integrating AI into this imaging workflow can standardize the measurement. That is very important. Standardize the measurement and also stratify the risk and personalize care. So hoping for collaboration between engineers and clinicians that will definitely drive innovation in reproductive health. So these are my few references in last 5 years. Studies have been done, is also going on. Hope for the future the best result will come. Thank you.
Thank you, ma'am. Actually, very sorry once again. So when I met her, she was so energetic. I thought that she is doing her MD here only. So
Thank you, sir, for the compliment.
Sagarika, ma'am. So
Yes, sir.
Sagarika, ma'am, working for working experience is around 8 years. She has already completed MBBS and MS, and she is mainly known for his high risk pregnancy management, and she also trained herself as a gynecology laparoscopic surgery, and mainly holds a special interest on this adolescent health and preventive oncology. So once again, very sorry, ma'am, I have not introduced.
It's
I thought
fine,
that
sir.
you were so much energetic. I thought, Miss, in that day also when I met and they were discussing with Pamela, ma'am, I thought that you are as a MD student. Very sorry once again, I have not.
No, this is fine, sir. Thank you, sir. Thank you.
So I think you have already given so many problem statement very clearly what we can do, what will be the proposal, what are the different problem statement we can work together. So definitely I think after this I'll meet you and we can have a lot of problem statements in which we can
Yes.
engage our faculty and students for the future research scope.
Yes,
Now
sir.
I'll open to the audience. So if you have any questions, you can unmute yourself one by one. You can ask. Yes, ma'am. Yes, students, you can participate. You can ask. Yes. Any participant having any questions? You can unmute yourself. Ma'am, here we have Dr. Punabe is here. So who is also working as assistant surgeon, but he is so much interested about this AI. Now currently he is doing his MCA in AI and data science. He is also here. And Dr. Punapa, any suggestion, any question from your side?
Uh, I don't have as such any questions because it's a little out of zone from my surgery department. But one thing I have noticed is there are a lot of infertility cases coming up these days in the modern girls. And most of them, which I have noticed, is no menstrual irregularities, no obesity, no metabolic complications, and no hypoandrogen. And EMH, prolactin, everything is normal. So I think there is still scope for developing this AI model, which you have preliminarily started only on the image. I know this conference is mostly on the imaging part of it, and you have presented exceptionally well in that ultrasound ovary and parameters and all those things. But to really look into infertility, I would appreciate maybe we can get some more data on all the metabolic parameters and all those things and see where actually we are going wrong. Maybe this is just a suggestion to develop on the model which you have you're trying to build up. Thank you.
Thank you, Punapa. So now next, Yatharth. Yes, any question from you?
Hello, ma'am. Am I
Hello.
audible?
Yes, yes, you're
Yes.
audible.
My question was that, as far as I know, PCOS does not really have a cure or like a surgery that can cure it. It's more of a you have to maintain it for the rest of your life and keep it under control. While PCOD is a more milder form of it, which happens due to slight imbalance, and a lot of the times I think PCOD is what evolves into PCOS later down the line. So should we be focusing on diagnosing PCOD instead of PCOS because that has a higher chance of like being able to reverse it while it's in its early stage and before it evolves into a lifelong sentence?
Yes, definitely. PCOS is a syndrome that is a clinical constellation of the symptoms arising out of all these hormone imbalances. So the main treatment is lifestyle modification only. Like you have to do regular exercise, yoga. Obesity should be avoided. So weight reduction is the first basic step we have to do, and diet modifications. Avoid all oily foods, and the diet should be specified, and with this life modification, most of the patients improve clinically. So, but for diagnosis, as per the Rotterdam criteria, for diagnosis purpose, we need to do ultrasound and some blood test investigations. Like it's a combination of clinical and the diagnostic procedures. So 2 out of 3 should be present. So some patients are there, there will be no menstrual irregularities, no metabolic factors, but in ultrasound, there will be polycystic ovary will be coming. So the HRE 223 guidelines, they have specifically mentioned what is the specific diagnostic findings in PCOS, like number of follicles per ovary, number of follicles per section of ovary, what is the Doppler indices, what are the echogenicity of the ovarian stroma. All this should be taken into account in case of PCOM type of PCOS. That is polycystic ovarian morphology type of PCOS. We have to go for this imaging.
I see. I have another question related to that. It's not really a technical question, but uh, as I was saying that the earlier the diagnosis is done, the better. And a
Yes.
lot of young teens and like at least matlab, around 18, 19-year-olds are also getting PCOS and PCOD. So how do you think we break this societal taboo thing that we don't have to get diagnosis until it gets worse? Because I have seen this a lot in, like, no offense, but I've seen this in Indians that we as people we don't tend to go to the doctor until it gets out of control, or it pains a lot, only then we'll go. So how do you think we tackle this and actually promote the diagnosis? What if we invent a tool and no one even wants to use it? So how do we change that mentality as a society, according to you?
Yeah, basically it needs awareness only. Awareness in the society. Through various platforms, we can aware the young adults to basically modify the lifestyle, because these days all are just crazy for all these fast foods. They are not going for exercise or yoga, and stressful life they are having. Also delayed marriage, that is also factor for this infertility issues. By the time after 35 plus, as such, the follicle, the ovarian reserve will be less only. So definitely there will be infertility issues. So they are more career oriented. That's a good thing, but we should age of the marriage should not be delayed. Okay, so there are so many platforms like PCOS societies also is there in our India. So they are conducting many awareness programs, and through social media also we can spread the awareness that what is PCOS, how it can be prevented or it can be treated by the first and foremost changing the lifestyle. We should avoid the sedentary lifestyle. At least 3 to 5 days in a week, we should go for a mild walk or normal exercise we can do. At least yoga, that's the best thing we can do. We should take few, at least half an hour in a day for our health only. So from the starting, from the teenage also starting from the family, in our education system, through social media, we can definitely create awareness in the society.
Understood. Thank you.
Thank you.
Yes, Soumya, next. Yeah.
Good morning, ma'am.
Good
So my
morning.
question is like you have in your field you are studying, it is a multifactorial conditions, like when it has so many factors to be monitored. So the single integration of any AI or an LLM will not give the accurate result. So how can we overcome, like we are doing we have to do multimodal things, which is a bit hectic. So what are the procedure we can follow to simplify the procedure, if any, please? Uh,
Yeah, definitely it's a multifactorial disease. So three factors as we have discussed, like first one is clinical, like menstrual irregularity, whether she is having any symptoms of hyperandrogenism, basically acne or hirsutism. These few points must we have to take for the diagnosis. Then we'll go for imaging like ultrasound. At single ultrasound we have to do at least. And third, basic blood tests like sugar level, the lipid profile. So these few tests we have to incorporate in the model. By just by assimilating all these factors only, we can conclude or we can predict that the patient is having PCOS. It is in the diagnostic criteria only. So basic these 3 points we have to take into consideration.
Thank you. Sohini, last question from you, then we'll end. Yes, Sohini.
Good morning, ma'am. Thank you so much for the session. Am I audible?
Yes,
Yes. Good morning.
So, ma'am, actually, I'm a student from KIT and I'm currently pursuing my MTech in biotechnology. And I'm currently 23 years of age. I'll finish the next year. So, ma'am, I have a question. As a female who is 23 years old, I have never had any problems related to my menstrual cycle like PCOS or PCOD. So in this case where I already have a normal cycle going on, are there any chances due to some lifestyle changes to develop these conditions later down the stage in my career? Like let's say if I have a very packed up schedule, let's say I'm standing in lab for long hours and my lifestyle becomes like that only. It's more or less like a sedentary lifestyle with very least amount of activities. So given that I have no history of PCOS or PCOD as such till now, are there possibilities or chances where I might as well develop these conditions? And if so, like what preventive measures should I start taking from now? Because I already do have a very sedentary lifestyle.
Yes, it's a disease in starting from the adolescent through the reproductive age group. In case of postmenopausal lady also we find PCOS, but all patients will not have the similar features. Some will have menstrual irregularities, some will have nothing, but in ultrasound there will be PCOS. Some will develop some metabolic complications in the later part. So at any age group it can develop. So I will suggest that we should, as we have already discussed, the sedentary lifestyle we have to avoid. We should take out at least half an hour in a day for exercise, maintain our BMI, maintain our weight as per our height and weight and age. And all these junk foods, all this should be avoided. And one thing is, there is also genetic predisposition for this disease, like in family members, mother is having, sister is having, you may develop. So from precaution, we can take the basic is lifestyle modification only. And if you are having some menstrual irregularities or any complication, definitely you have to consult the doctor, and the treatment should be started as early as possible before going for any complications. Main thing is lifestyle modification only.
Okay, sure, ma'am. Thank you. And ma'am, does a diet play a very important role in this case? Because many
Yeah.
people, ma'am, personally, I'm a vegetarian. So, like, will that have any significant effect on this with age, or is it just sufficient nutrients could be enough irrespective of the diet?
No, diet means non-veg should be, excess of non-veg should be avoided. In vegetarian also it can develop if we will not maintain our BMI, our weight as per height and age. If we don't maintain it, definitely it may lead to metabolic complications, like your lipid profile may be deranged. So routinely we have to screen for that. At least in a year we can screen for lipid profile, what is our sugar level, all that. And we have to monitor our body. So, and avoid all kind of junk foods and heavy oil. All this we have to avoid definitely, whether vegetarian or non-vegetarian. So we can definitely modify.
Okay, sure, ma'am. Thank you so much for attending my query.
Thank you.
So thank you very much, ma'am. Definitely, I'll be in contact with you, and very informative session this one is. Thank you very much.
Thank you so much, sir. Thank you.